Okay, let's start. Good morning, everybody. It's a great pleasure and a great honor for me to start and introduce one of the highlights of our meeting here. And I will chair this session together with Helena Prisby from Gothenburg. My name is Hans-Joachim Wilke from Ulm in Germany. And we are very proud to hand over the ISSLS Prizes now. The ISSLS Prizes are sponsored by the European Spine Journal since a few years. Helena will talk about this briefly. And I would especially like to acknowledge the committee which makes the selection process. There are so many papers which are submitted for this prize because it's such a prestigious award, probably one of the most prestigious awards in spinal research. And you can imagine it's a lot of work, and everybody of this committee is reading each of the papers, evaluating them, and then they have discussions how they want to decide. And you see, these are the most of the most important people here from our society. It is our current president, Kazushiro Shiba, the president which will be the president tomorrow, Peter Robertson, then Tim Yoon, the immediate past president, Daisuke Sakai, the secretary, and another upcoming president, Jeff Lotz, together with Kazushira Takahashi and Helena Prisby, actually the chairwoman. And this is the right time to hand over to her, and she will introduce the awards. Thank you.
Thank you. So the history of this prize was that it began in 1981. Then ISSLS established an annual prize to recognize outstanding contributions to the field of lumbar spine research. And initially, as most of you know, it was named the Volvo Award, and then it became the ISSLS Prize. So there is 2 to 4 papers that have been awarded annually ever since. And over the years, this adds up to about 150 papers. And these have been in the categories of clinical, basic, bioengineering, science of the lumbar spine. And many of the papers here that have been awarded the prize have become highly cited papers. So this year, there were 22 papers submitted. And this is a slightly decrease compared to the 2 last years. So I would like to take the time to encourage everyone to send your best papers to this award to apply for the prize. And these were, as usual, in the categories basic, clinical, bioengineering, science, or combinations. And the committee this year decided to award 2 prizes, and they do receive the honor, but also some monetary encouragement. The papers are submitted to the European Spine Journal and also to the ISSLS at the same time. And the committee, as we were told, evaluate all the papers, every member of the committee, but also we have access to the evaluations, the review from the reviewers in the European Spine Journal. This means that it's between 7 to 10 evaluators for each manuscript. After evaluating them, we had a final Zoom meeting in January 2024 to discuss the manuscripts and decide about the winners. Of course, we declare interest of conflict, and if there is someone on any of the papers, they don't participate in the final decisions. The 2 winning papers are, as we know, published in the European Spine Journal this year, and all of the other papers are also considered for publication in the European Spine Journal. Of course, we want to acknowledge European Spine Journal, represented by Robert Gunsberg, Editor-in-Chief, who make this possible for us to give out this prize. So thank you. So we move on to the important part, the award ceremony and the paper presentations. First, we will announce the papers, and I will ask the members of all the authors of the papers to come up, and we have the ceremony. After that, we will have the presentations of each of the papers, and of course, discussion. So take the chance to ask them questions. So, the first paper in the category Basic Science of the ISSLS Prize 2000 and 2024 has the title, Superiority of Nucleus Pulposus Cell versus Mesenchymal Stromal Cell-Derived Extracellular Vesicles in Attenuating Degeneration and Alleviating Pain. And there is a lot of authors, and I apologize for if I'm not pronounced the names correct, but the authors are Ambrosio, Scholl, Ruiz Fernandez, Tamagawa, Soma, Tilota, Di Giacomo, Sisone, Nakayama, Kamika, Papilia, Sato, Vadala, Watanabe, Denaro, and Sakai. So please, all of the authors that are here, please come up to the stage. And we need to have you somewhere where we can have the photo. To go down there, maybe? So the second prize for this year is in the category Clinical and Bioengineering Science. And the title is How Standing and Supine Positions Influence Nutrient Transport in Human Lumbar Intervertebral Discs, a serial post-contrast MRI study evaluating interplay between convection and diffusion. And the authors are Naresh-Babu, Jajendra, and Praival. Please come up to the stage. Congratulations.
Thank you. Thank you.
So we can take some photos again. So I now want to ask the author of the first paper to come up and present. Dr. Ambrosio, please.
Thank you very much for the kind introduction. It's such an honor to be here to give this talk today. So as we all know, intervertebral disc degeneration is characterized by the progressive loss of nucleus pulposus cells and the breakdown of the extracellular matrix, eventually leading to major structural and biomechanical alterations. Therefore, replenishment of defective cells with potent stem cells has been long considered as a possible solution to this huge issue, but does not come without downsides. These include poor cost effectiveness and, of course, the doubtful survival of cells once injected into the harsh degenerative environment of the disc. And as an alternative solution to stem cells, local progenitors hallmarked by the expression of the Tie2 receptor have been also explored as a possible alternative. Meanwhile, we have been witnessing the increasing popularity of extracellular vesicles in the field of regenerative medicine, with several applications also described in our field. These mainly came from MSC-derived EVs, while less is known regarding EVs isolated from healthy MPCs and their progenitors. So actually, we asked ourselves whether EVs isolated from healthy MPCs could actually possess intrinsic regenerative capacity, and if that capacity could be associated with the expression of the Tie2 receptor. And we also questioned ourselves whether it was possible to manipulate MPC culture conditions to optimize EV cargo and yield. So in the first branch of our study, we isolated MPCs from young and healthy donors, and actually these cells underwent two different culture protocol. One was a standard protocol which adhered to the recommendations of the ORS spine section, and another one was a specifically designed and previously described Tie2 enhancing protocol. Then we also had a commercial line of bone marrow derived mesenchymal stromal cells. All these cells will culture expand into D, conditioned media were collected, and EV were isolated with ultracentrifugation. Then, according to the recommendations of the International Society for Extracellular Vesicles, we performed thorough EV characterization through Western blot, transmission electronic microscopy, Q-nano, and we also assessed EV uptake within recipient MP cells under confocal microscopy. Then we isolated MPCs from degenerative specimens of patients undergoing lumbar spine surgery. We culture expanded them into D, and we treated them with either EVs isolated from MPCs undergoing the standard protocol, which I will call standard EVs, and EVs isolated from MPCs undergoing the Tie2 enhancing protocol, which I will call Tie2 EVs. And then we checked for cell proliferation and cell senescence. As you can see from our characterization experiments, the TEM analysis, our Q-nano analysis, and also Western blot confirmed that morphology, average size, and marker expression actually confirmed the EV nature of our nanoparticles, which was really close to the exosome class of EVs. Then when we checked for confocal microscopy, we basically labeled EVs with PKH26, and we incubated EVs with stained MPCs, and at 3 hours, you can really see that these EVs gather around the nuclei and are actually taken up by the cells. Then in terms of cell proliferation, we tried to test 4 different concentrations of our EV product according to what we found in the literature, with and without interleukin-1 beta to mimic the inflammatory environment of the disc. First, we could not see any major change among the 4 tested concentration, and that's why we chose to proceed with the intermediate one in our subsequent experiments. And then we could also see that while standard EVs did not significantly increase cell proliferation at both time points, Tie2 EVs significantly increased the number of proliferating cells, also in presence of interleukin-1 beta. In terms of cell senescence, we could clearly see that interleukin-1 beta increased the amount of senescent cells, while both standard and Tie2 EVs reduced the amount of senescent cells. And the latter were also able to blunt the pro-senescent effect of interleukin-1 beta. Indeed, you can see here that cells treated with both Tie2 EVs and interleukin-1 beta have significantly less senescent cells compared to interleukin-1 beta treated cells. Then we went on in our in-vivo study, and basically we employed a rat model of intervertebral disc degeneration, namely the annular tail puncture model. We had 16 animals divided in 4 groups. All these animals were injected at 3 different levels at the coccygeal discs. And actually we had a sham group injected with PBS, and 3 groups injected with EVs. One group was injected with standard EVs, another group was injected with Tie2 EVs, and the last one was injected with BMMSC EVs. All these animals underwent biweekly pain behavior assessment, monthly disc height index measurement, monthly IL-6 serum concentration assessment, and post-sacrifice we checked for gross anatomy and histology. You can see here the von Frey test that actually animals stimulated at both the surgical side and tail-based showed a different response, and this was actually a test made to assess the pain mechanical sensitivity of the animals. Since week 2 until the end of the animal's lifespan, we could see that both animals treated with standard and Type 2 EVs, with the latter being the most effective ones, consistently show the highest pain threshold, while animals treated with both sham and BMSE EVs did not show significant difference among them. And this was consistent at both the surgical site, which was right where the injury was made, and at the tail base, which was just proximal to that. Then we also performed the open field test, and we used a really high-end software which was able to track the animal with AI and extract dozens of different outcomes, such as mean velocity, total distance, probability of grooming, sniffing, and all that. And these are all behaviors that actually reflect the pain, anxiety, and depression state of the animals. But while we found some isolated significant changes, there were no substantial differences among the groups. When we checked for the IL-6 serum concentration, we could see that animals treated with Type 2 EVs had the lowest level of these circulating cytokine, although these differences were not statistically significant. Then when we checked for this guide, we could see that in the sham group, we had a progressively decreasing height of the disc as expected. In the BMSE treated group, we had an initial decrease and a later increase, even if this was not statistically significant, while we were really happy to see that animals treated with both standard and Type 2 EVs maintained their disc height. And this was later confirmed by our macroscopic and microscopic assessments. You can see here the macroscopic photographs that actually animals treated with both EVs showed a preservation of disc height and a clear distinction between the AF and the MP, and this was also reflected by our histology and, of course, by the semi-qualitative assessment of both macro and micro imaging with the Thompson score and the RS spine score. Both standard and Type 2 EVs perform significantly better than the other two groups. So we may conclude by saying that probably EVs isolated from healthy and young NPCs may constitute a promising tool to regenerate the IVD in the future. However, we really need to further characterize what's inside these EVs and how they work, and that's what we are doing right now as a follow-up project. We should also acknowledge that this whole field is affected by a multi-level variability, which really is a great issue that should be acknowledged at a national and international level. So I would like to acknowledge all the people who made this possible, my mentors, Professor Valalain, Professor Daiske, all our funding bodies, and all of you for the attention. Thank you very much.
Dear Luca, thank you very much for this very nice presentation. The floor is open for discussion. We decided that we discuss each paper separately. Yes, please.
Stefan Dudley, University of Zurich. Wonderful project. I was wondering about immune rejection of EVs from NPCs. So do they express MHC molecules, the NP, and are they present on EVs? Because if you want to inject them in that generated disc, we probably also have presence of immune cells.
Yeah, thank you, Stefan, for the great question. This was actually one thing that was raised by one of the reviewers. So there is evidence that these EVs do not express that much of MHC. And actually, all the previous reports on xenogenic and transplantation of different EV products across different species did not report any immunogenic reaction. And actually, in all our samples, we did not find any of those. So we can reliably affirm that maybe that's not an issue. So we did not encounter any, kind of that reaction, and also previous paper did not report any of those. So I think that it's not an issue.
Okay, thanks.
Thank you. All the top specialists are standing on the microphone. Ben Gantenbein.
Thank you. Luca, very nice work. I have two questions, actually. So one, it looked like your EVs from the type 2 cells are a bit smaller than the other EVs. Do you have any explanation for that?
So actually, those were only representative images. If you see, and this information is also available in the paper, the smallest ones were the BMSC derived ones. So the average size of both healthy MPC and type 2, standard MPC and type 2 MPC derived EVs was around 109 nanometers, if I recall correctly, but this information is in the paper, while the BMSC derived ones were around 90. So both fall in the small EV class. I don't think that 10, 15 nanometer difference makes a huge difference in terms of biological activity. So they fall in the same class still. But I don't really know if that's a big difference. And now the new edition of the ISF guidelines really underline the need to perform biogenesis-related experiments to make an assumption regarding size, class of EVs, and their biological activity. So we need to delve more into those mechanisms to really give an answer to that.
And then another short question I have, what is your clinical perspective? So how many cells can you get out per donor, an average from type 2 cells, and how many cells, or how do you foresee that we can have enough extracellular vesicles to for a treatment of a human patient, for instance?
Yeah, thank you for your nice questions. That's something that I've been wondering a lot. No? So one thing that is really interesting regarding the whole EV field is that basically you use a byproduct, because that, when you change the media, if you don't use that for EV isolation, that media is gone. It's just trashed. So good thing is that I suppose that this whole possibility has a strong effect in terms of cost effectiveness on a manufacturing level. And plus, the idea would be if we are able to select the right donors and actually fine-tune the culture conditions that would allow us to have a stable EV throughput with the content we need and the final effect we need, I think that once that can be standardized, if that can be standardized, would be a huge hit also to put that on the market and maybe make a drug out of it. But it's really far.
Thank you.
Okay, the next question from Graciosa Teixeira.
Thank you. Congratulations, Luca. Very nice work. So my question is with regard of the mechanism of action. So bone marrow EVs from the MSCs are very often discussed to be immunomodulatory. But you've very nicely shown in your ex vivo data this accumulation of the EVs around the nucleus and the decrease of the senescence of the cells. Do you think the type 2 have a different mechanism of action? Can you speculate about that? Or maybe you have already some preliminary data?
Thank you for the nice question. So that was also one thing that really interested us at the beginning. So since we saw that type 2 EVs showed a slightly higher and better effect compared to standard EVs, we were initially really convinced that maybe type 2 expression was associated with a nicer and a more effective, final outcome on cells. But maybe, since both type of culture conditions are actually applied to the same cells, which are healthy and young MPCs, maybe it's probably that is more associated with what those cells are and the fact that they are really healthy and young more than associated with raw type 2 expression, because we all know that it's really difficult to capture type 2 expression, and that's really labile and can change across different passages. So right now we are concentrating more on the lineage and more on what those cells are rather than on raw type 2 expression.
Yeah, because if I may follow up on that and the discussion regarding the clinical treatment. So if you go and try to do sorting for type 2 cells, this makes the whole process more expensive and more time consuming. So if you can indeed show that without this additional step, it would be very valuable.
Graciosa, maybe you can discuss that
later.
Yeah,
thank you.
I would like to allow the last question to Mariana Trifonidou.
Thank you.
Then I will ask my most important question. I have two, but I will do only one. So you claim that the effects that you see are EV associated. How can you prove that? Because we know that with ultracentrifugation there are a lot of cowbell isolates. Did you take EV depleted controls with you?
So that is, one of the biggest issues that actually affect the whole field. So what we did was when we isolated EVs, we used EV-free FBS, and one thing that we tried to do for sure was...
I don't refer to FBS, but ultracentrifugation spins down big protein complexes, EVs, and all the other stuff. So you can't really say it's EV associated. It can be simply other factors have been secreted.
Yeah, no, in that sense, we did not, do anything specific related to isolate pure EV-related effect with co-associated precipitated material.
Thank you.
You see, this was the right paper. Very hot topic. Thank you so much.
Thank you.
And we come to the next paper.
So then we ask the winner of the second prize, Dr. Naresh-Babu, please come up and give your presentation.
Good morning. At the outset, I would like to thank the ISSLS Award Committee for recognizing our work and being selected for the prize. And I'll bring greetings from India. So our work is on how standing and supine positions influence nutrient transport in human lumbar discs. It's a serial post-contrast MRI study evaluating the interplay between the convection and diffusion of solutes across the disc. So this study received grants from Spine as a part of Spine startup research grant for the study. We all know that intervertebral discs and nutrition is a topic which is mostly discussed in many of ISSLS meetings, because disc being the largest avascular structure, the nutrition is the final common pathway for disc degeneration. And also, the metabolic and solute transports in and out of the cells, they maintain the equilibrium of the solutes and metabolites inside the disc. So measuring solutes transport has been a problem because there are many attempts to measure the solute transport into the disc by doing radioactivity in harvested discs, and by a lot of studies done on in vitro models, and histological studies have been done, and there were attempts to put a oxygen introducer to find out the oxygen concentration inside the disc. But the monumental work done by Dr. Rajshekhar and along with us, so the serial post-contrast MRI studies have been a breakthrough in actually studying the disc nutrition diffusion across 24 hours in humans in a non-invasive way, in vivo. So this work has actually received ISSLS Prize in 2004 at Spine Week, Porto, 20 years back. So we continued our study, with also studying the annulus fibrosus diffusion by also doing the same kind of serial post-contrast MRI studies. But diffusion has been the predominant mechanism of nutrient transport across the disc, but disc nutrition is not only dependent on diffusion. There are two ways of solute transports across the disc. One is by process of diffusion and other is by convection. The diffusion based on the concentration gradients, and it's a passive transport of solutes across the disc, and small solutes are usually being transported by diffusion, and usually the glucose, oxygen, and lactate in and out of the cell are being done by diffusion process. But convection is a pressure gradient, so it requires a load-induced transport. Whenever the patient actually, whenever the individual stands, walks, or bends forwards, and there is application of the load, the process of convection actually takes place. And it's useful for the large molecules and solutes. Sometimes this can be forced into a forced convection. Usually glycosaminoglycans, growth factors, proteins, enzymes, and hormones are being transported by convection. But applying load has been a problem, and studying this load and also solute transport has always been a problem. So there were attempts to put external compression devices on animal models and study the serial MRIs, how the diffusion happens across the disc. And there were also attempts in the humans by applying a MRI-compatible loading device and subjecting the patients into serial post-contrast MRI studies and find out what happens as a part of diffusion and what happens as a part of convection for the solute transports. The purpose of our study is to actually study both transport mechanisms of diffusion and convection, natural physiological loads and conditions, in vivo, non-invasively in humans. This has been a big task, how to apply loading in a MRI, so in the natural loads and study these ones. So we strategically employed positional MRI imaging and capture the serial post-contrast MRIs over a 24-hour period under natural physiological loads. So we have an access to this positional MRI. This is a low-field magnet. We can see this entire MRI gantry along with the examination table can be tilted upright, and the patient can have a supine MRI as well as loading MRI. So we utilize this technology by which we can actually apply loading under natural physiological conditions and do serial post-contrast MRI studies as we do it in supine. So this is a 0.25 Tesla magnet, and this has got a foot plate wherein the patient can comfortably stand, and there is a strap put onto the chest, and the coils are already applied to the lumbar spine, and the patient and the volunteer actually can have a vertical shift. And then we done this study in supine position and also in 90 degrees upright position. And also we made the patient into recovery supine position and applied the same serial post-contrast MRI studies. So once the MRI is complete in supine, we ask the patient to come to the study in standing, and then again we made the patient to recovery position, wherein the patient goes back to the supine position and see how the loading and unloading of the disc happens in these patients. So this technology gave us the advantage of studying convection along with the diffusion in the humans. So we have done the study in supine, which acts as a baseline, and standing position for the effects of natural standing, and recovery for post-standing recovery. The serial post-contrast scan was taken after giving 0.3 millimoles per kg body weight of gadolinium, and at 2 hours, 6 hours, 12 hours, and 24 hours. A serial post-contrast MRIs were done, and then all the studies were done from 8 a.m. of the present day to the next 8 a.m. to account for the diurnal variation of the contrast. The MRI, as I mentioned, is a 0.25 open MRI system. It's a multi-channel spine coil, dedicated coil was used, and is rotated to 90 degrees. This is the MRI protocol by which we take these images. We had 100 healthy lumbar disc segments of Firman grade 1 included in the study from 20 healthy volunteers, 20 to 30 years of age. So each participant underwent 5 MRI examination in each position, that is supine, standing, and recovery position. And a total of 300 MRI sequences were obtained, which provided 1,500 disc segments for analysis. And the study consisted of measurement of 7,500 assay values, 4,500 in the disc and 3,000 in the end plates. So the regions of interest were studied according to the description by Dr. Rajshekharan et al. Pre and post contrast mid-sagittal T1 images were taken. Superior end plate, inferior end plate, central nucleus pulposus, superior nucleus pulposus and inferior nucleus pulposus were measured by putting a manual cursors onto the disc serially at each time intervals. So baseline values were calculated, and the signal intensity at particular time interval was calculated. Then enhancement percentage were calculated by using the formula, and the time intensity curve was plotted basing on the time and the interest. So these are the images how we got the patients. This is one disc of 20 patients at 3 different time intervals. You can see, you can still appreciate some amount of bands which are seen on the post-contrast MRI images, but exact measurement of these band signal intensity will give us exact values of how the diffusion happens in different parts of the disc. So this is a busy slide where you have a lot of values. So then I'll go into what happens when the patient is in the supine position, regular diffusion process. We all know that end plate takes some time to enhance and it reaches 2 hours to reach the peak and then gradually drops down over a period of 24 hours. While the disc takes some time to reach and reaches peak at 6 hours and then gradually drops. This is the normal diffusion pattern which we have published in 2004 based on our largest human study sample of serial post-contrast MRI. So once we applied 3 loading positions, what exactly happened to these patients, we can appreciate this is in the central nucleus pulposus. When the patient is in supine, there is an increase in contrast enhancement. But when patient attained a standing posture, you can see sudden dip in the loss of enhancement. And then when the patient again resumes recovery position, there is again regain of solutes happening across the disc. That has been seen in all time intervals and predominantly in the 6 hours and 12 hours. We can see whenever the patient stands, there is a drop in the enhancement, and when the patient takes relaxed position and recovery position, there is an increase of enhancement. So that's the graph. So the standing causes depletion of nutrients, and the recovery position actually increases the regain of the nutrients. Then we applied the end plate and also peripheral nucleus pulposus. You can see whenever there is an increase in enhancement of the red one is peripheral nucleus pulposus and the blue one is end plate. And you can see whenever the patient takes a standing position, there is a drop in enhancement of peripheral nucleus pulposus. At the same time, there is increase in enhancement which is taken up in the end plate. This is very interesting, which we couldn't find, which we didn't expect to happen in the patient is standing. And this process almost continued the entire process. Whenever there is a increase in the disc diffusion in the central nucleus pulposus, and there is a peripheral nucleus pulposus, there is depletion in the end plate, and vice versa. So this is the central nucleus pulposus graph, and you can see the peripheral nucleus pulposus also followed the central nucleus pulposus, but the end plate has almost has a reversal. Whenever there is increase, there is a depletion. So effect of standing on nucleus pulposus, there is a sharp dip when the patient assumes a standing position of 40% at 2 hours, 61% at 6 hours, 84% at 12 hours, except 24 hours, every time interval there was a drop. And peripheral nucleus pulposus and inferior also followed the same pattern. But in the end plate zone, there is an immediate increase of enhancement when the patient attains a standing position. So superior end plate and inferior end plate has different values of the amount of increase in enhancement. So whenever there is a loss in peripheral nucleus pulposus enhancement, there is a gain in the superior corresponding end plate enhancement. That is interesting finding which we observed in this study. What we try to say is there is an average of 26% loss of central nucleus pulposus enhancement on standing, and superior nucleus pulposus lost 175%, and superior end plate gained 105%. At the same time, inferior nucleus pulposus lost 96%, and inferior end plate gained 90% of enhancement, suggesting there is a contrast match which is happening from the central nucleus pulposus to the peripheral nucleus pulposus to the end plate whenever the loading is applied. So what happens when the patient actually assumes a post-standing recovery and in the supine position in the healthy discs? The end plate lost the average of 90%. Whatever the enhancement which was gained during standing was lost, and peripheral nucleus pulposus recovered the enhancement. So the amount of loss in end plate zone was 900%, and the amount of peripheral nucleus pulposus recovery was an average of 920%. So, and also it's interesting to note that the nucleus pulposus and peripheral nucleus pulposus recovery surpassed that of a baseline value. So standing versus recovery, standing acutely squeezed out an average of 26% of solute small molecules out of the central nucleus pulposus by washing out phenomena. And immediate post-standing recovery position sucked in the average of 104% of small molecules into the central nucleus pulposus. There is a pumping in phenomena post-standing recovery. So diagrammatic representation of the same thing. You can see the base value in the supine. There are the solutes are slowly diffusing into the disc. And you can see in the standing, there is a depletion of solutes going out of the disc. And then in the recovery, you can see, again, there is a infusion of solutes. You can observe in the magnified view, you can see in the 2 hours, there is a contrast enhancement, and standing, there is a depletion, and then again, recovery, you can see an increase in the There is also regional differences which we notice in superior versus inferior, and superior being more effective than the inferior nuclear pulposus. There are temporal variations also seen across the time. The depleting effect increased with the time, but whereas the recovery is gradient dependent, depending on the amount of solute concentrations it happened. The rapid effect of short loading and unloading, there is a dynamic interplay between the diffusion and convection, and this highlights the disk's ability to swiftly respond to the changing concentrations of the disk. The limitations of our study being a low field magnet, there is a manual placement of the cursors, and there may be potential minor changes of volume effects because of manual placement. But we use larger cursors, and induced changes may be similarly affected in all the intervals, and also variations in positioning of the patient from supine to standing may have different problems. So to conclude, to our knowledge, this is the first human in vivo study which non-invasively evaluated the effect of diurnal natural physiological loads on solute transports across the disc. Standing causes rapid depletion of the solutes by squeezing out phenomena. Immediate post-standing recovery results in sucking in phenomena. And during rest, solutes diffuse gradually into the disc by diurnal, and short loadings, unloading redistributed the small solutes by forced convection. So thank you very much for your attention. I would like to acknowledge my sincere thanks to my mentor, who has instrumental to introduce me into the ISSLS, into this wonderful committee. And also I'm very thankful to my family for sparing time and giving us encouragement to do this stage. Thank you very much for that.
We will steal a few minutes from the coffee break because we need to discuss this. So please introduce yourself and ask your questions. Should we start, Sharma?
Hi, Asim Sharma, University of Michigan. First of all, congratulations, wonderfully conceived study. I have two questions for you. One, you acknowledged in your limitations the placement of the ROIs. So it would have been nice to know how much variability that causes, and you could test that by repeating the measurements on the same scan. Have you done that?
Yeah, this is actually done in the initial study, which once we have done that. So we identified that when we place the small cursors, the previous studies actually suggested to place 1 mm square cursors at different parts of the disc. But the partial volume effect can be mitigated if you use a larger cursors of a bigger square area. So that averages the amount of issues. That has been done with...
that is correct, but
yeah.
Since you're comparing two different scans and you're essentially saying that there is 120% enhancement compared to the other scan, I think it's important to know that in this study, how much variability in the numbers can we get? So that's something to consider. The other question I have is that with MRI as opposed to CT scan, the signal intensity values are not calibrated, which means that when you take two scans, you can get completely different signal intensity values once you draw the regions of interest. So how did you correct for that?
Yeah. So two things. One is if you take two different scans, then we'll find the different signal intensities. But what we have done is we are comparing the same patient, same scan, same mission at different time intervals by giving the contrast, and we are trying to measure the contrast from the baseline.
No, so but you're scanning the patient at different times.
Yes, of course. Yeah.
So even if it's the same machine, you can't just assume that they will give you the same numbers.
Yeah, so what we made sure that we obtain the mid-sagittal cuts of all the patients and measure in the mid-sagittal cut. I understand that there has been a problem, that's one of the limitations, but to see that the amount of change of happening 900% can be considered like.
I think you have to continue
okay.
with this. We have so many, and we have. We'll allow a few more questions, please.
Hello, Keita Ito from Eindhoven and Utrecht. Congratulations on a very nice study. It's very interesting data. I was wondering, based from your conclusions, are you concluding that your data supports that convection is actually helping to pump into the disc from the vasculature this gadodiamide? Is that the conclusion drawing? Well,
yes,
I would like to propose another sort of conclusion. If I look at your up and down data, it looks like that you're getting a redistribution from your NP into the end plate and back into the NP. But that actually, if you look over time, both in the end plate and the NP, you see sort of a decline, right, in the concentration. And I don't know what it looks like in the signal inside the vasculature, but you probably also have a very similar decline in the signal, which would seem to indicate that it's still a diffusive process from the vasculature into the disc, but that the convection transport that you're seeing is actually just within the disc. How would you respond to that?
Yeah, so thanks for the wonderful observation which you have noticed. So we have also discussed the same thing while we are analyzing the results. So we completely agree with you that the convection actually redistributes the solutes into and out of the disc. So what we try to say is whenever there is loss in the nucleus pulposus, where the disc solutes are actually leaving out of the disc, whether they are going into annulus, whether they are getting absorbed into the disc, or they're going to the end plates. What we proved in the study is actually the solutes are actually moving out of the disc towards the end plate zone. But I can't say it's end plate, say end plate zone, that area. So, and then again, when the patient resumes the recovery positions, they're actually trying to re-enter into the disc. So, yeah.
Okay, short question, short
maybe
answers. We have to stop soon, but please, short question.
Naresh Kumar, very good study. Is there any thing which we can derive about antibiotic treatment for discitis in these patients? Is the position going to matter, or?
Yeah, there are two or three clinical implications which we are looking at it. One is like preventional strategies if you want to make about the disc degeneration, and therapeutic strategies like trying to use some cells to regenerate the disc. What is the right time interval in the diurnal cycle? And what should be the patient should be doing after injecting the therapeutics into the cell, whether it be resting or loading or unloading. Maybe disc antibiotics also one of the issues, but antibiotics has a different scenario because they have charged particles, but this is uncharged particle.
Okay.
Please, short question.
Yes, hello, I'm Dr. Chip from South Korea, Seoul National University. I'm a radiologist, so I am very curious about the measurements. The first thing is that the most important thing I think is already addressed, that you should have to normalize your data before measuring. For example, we usually, if you're measuring at the T2 weighted images, it's important to normalize using another ROI crossing at the CSF signal, etc. So the second one, most important thing is that, did you measure the signal intensity on T2 weighted spin echo images?
No. So T2 weighted are only used to actually classify the discs into, according to Fermat's grading. All the measurements were done on T1,
T1 weighted images.
Only T1.
Okay.
T1 and pre and post contrast.
Yeah, because you were showing on everything on T2 weighted images, so it was very confusing for me because usually gadolinium shows decreased signal intensity after on T2 weighted images. Okay, thank you.
The disc hydration, what you're seeing is not a hydrated disc, it's a diffusion of contrast into the disc.
Okay, last question.
Thanks, Aaron Field, San Francisco. Great study. To help support the conclusion that what you're seeing suggests that convection is important, I'm wondering if you looked at the rate at which signal intensity increased during inflow versus outflow. Some nice work by Keita Ito showed that flow rates are different going into the disc than they are out of the disc. And if it really is a convection phenomena, you'd expect to see differences in the rate inflow versus outflow.
Yeah. So that's also a wonderful question. But what we have studied is a static loading in two different positions. But the convection which is happening in static load might be different in dynamic loading, and the patient is really walking, bending, and doing daily activities. But what we really observed in this patient, in this population, is there is a redistribution of solutes, and convection really helps to pump in the small solutes also into the disc for a short period of loading and unloading.
I think we have to continue the discussion over coffee.
Thank you.
I would like to conclude this session. I would like to thank all participants, the authors of the prize paper, everybody who sent in their manuscripts, and again, encourage you to send your best papers to this award next year. Thank you, and a big applause for the two prize-winning papers.